Hello everyone and welcome to Instant Biology by Dr. Nila. Today I am going to deal with a very important topic that is very commonly asked in different life science examinations, and the topic that I am going to deal today is nucleotide excision repair. Now this topic comes under the heading DNA repair system in the molecular biology. For those of you who understand Hindi better, I have already made a video on nucleotide excision repair in Hindi. So you can watch that. And for those of you who understand English, so I am going to teach over here about nucleotide excision repair. Now before going into the details, let me first of all tell you what all we would be going through in this particular lecture. So firstly, I would be telling you about the introduction about nucleotide excision repair. That means what it is, why is it used, the mechanism behind it, and also we would be dealing with some of the questions that have been asked in different life science examinations related to the nucleotide excision repair concept. Remember, we have already discussed about the direct repair system, and we have already discussed about the one of the indirect repair systems also, in which we had talked about the base excision repair in English. Right? So in today's lecture, let us start nucleotide excision repair. So first thing first, what is nucleotide excision repair? Now, since I have already posted a video on the base excision repair, so it is somewhat, this nucleotide excision repair is somewhat very similar to the base excision repair only. I would be telling you about the differences that this system has with base excision repair, but primarily you can understand that the phenomena is quite similar. So the first point that you can see is it is similar to the base excision repair. So let me just quickly write it down: base excision repair. So nucleotide excision repair is quite similar to the base excision repair. This is the first point. And what we did in the base excision repair, you might be remembering if you have seen my previous lecture. If you have not seen, I would request you to go and check that out. So in base excision repair, what we used to do is DNA glycosylase enzyme was used in order to cut the glycosidic bond that was linking the sugar with the nitrogenous base. So this was the sugar and this was the nitrogenous base. The bond that was connecting the sugar and the nitrogenous base, that is called as the glycosidic bond, was cut off by DNA glycosylase enzyme. This was the first step in the base excision repair. But the process would be somewhat similar, but exactly it would not be the same. I would be telling you what we would be doing. Now, not preceded by the removal of the damaged base. So this is the major difference between the base excision repair and the nucleotide excision repair. What is the difference? In case of base excision repair, as I've already said, base excision repair, in base excision repair, we removed the damaged base. That means the damaged nitrogenous base. We are talking about the damaged nitrogenous base over here. What are nitrogenous bases? A, T, G and C. So whichever nitrogenous base was damaged, that was removed by cleaving off the glycosidic bond. By cleaving glycosidic bond. So this was done in case of base excision repair, but in nucleotide excision repair you would not do this. In nucleotide excision repair, the glycosidic bond is not broken and the damaged base is not removed initially, just like in base excision repair. This was the phenomena of base excision repair, but in nucleotide excision repair this process is not done. What is done? We would be seeing that in just a few minutes. Now it can act on more damaged areas. Now if I talk about nucleotide excision repair, it is used by the system, it is used by the body where the damage or where the intensity of the damage is very high, where a lot of damage is there. So in that case, the nucleotide excision repair is used. Primarily, it is used when a chemical modifier or a chemical agent acts upon DNA and it causes some problem in the DNA. Apart from that, pyrimidine dimers are also dealt with by nucleotide excision repair. Okay, so whenever a chemical adduct, whenever a chemical mutagen is acting on the DNA and it is creating problems, then this kind of nucleotide excision repair is utilized. Again, when the intensity of the damage is high, then nucleotide excision repair is used. Base excision repair is used when the intensity is not that much. Now, let us talk. So this was basically the introductory remarks, introduction of the nucleotide excision repair. Remember that, let me just add one more point over here, that this nucleotide excision repair would be used when there is a chemical mutagen. It forms adduct with the DNA, adducts with DNA. That means it will create some problem binding to the DNA, and then this kind of nucleotide excision repair will be used. Now, coming to the process, how is the nucleotide excision repair functioning? So the first thing is breaking of the phosphodiester bond on either side of the lesion on the same strand. Let us understand this. So what is the phosphodiester bond? Let us first of all understand what is this phosphodiester bond. So we already know phosphodiester bonds are the bonds between the sugar and the phosphate. Okay, the sugar and the phosphate have a bond between them, and basically that is called as the phosphodiester bond. Okay, the breaking of the phosphodiester bond on either side of the lesion on the same strand. Now let me just draw it in order to make you understand. Now suppose this is the correct strand. Okay, this is the correct strand. Let me draw the faulty strand over here. So suppose this is a faulty strand. Okay, so this over here is the faulty strand, and actually this is the problematic area. Okay, so this is the, let us say this is the lesion that we are talking about. Lesion is the problematic area. This is the lesion that we are talking about. It is the problematic area. So obviously you know that sugar and phosphate, basically sugar is connected to the phosphate, and phosphodiester bond basically connects the nucleotides together. So this would be one nucleotide, second nucleotide, third nucleotide. They are connected with the sugar phosphate backbone. What happens is the sugar phosphate backbone is broken down, or the phosphodiester bond is broken down on either side of the lesion. Let me say that this is one side of the lesion, this is one side of the lesion, and this is another side of the lesion. So that means we would be breaking the bonds present over here. So what we will be left with, let me just draw like this. So something like this we would be left with. Okay, so this would be what is left with, and this would be the original strand, the larger strand that we were looking at. So you can see that the bonds have been broken over here. There is no bond that is present over here. That means the sugar phosphate bond has been broken. So breaking of the phosphodiester bond on either side of the lesion. So this was lesion, on both the sides of the lesion the sugar phosphate bond or phosphodiester bond was broken on the same strand. So this was the problematic strand. On this strand only the bonds have been broken. So I believe you have got a bit of idea that how this nucleotide excision repair starts, or what is the introduction behind this. Just take a quick photograph of this and then we can move forward. Okay, so I hope you have taken a photograph. Let me move forward with this. So next is the second point. Is the first point was the lesion was present and the phosphodiester bond on both the sides of the lesion was broken. This was the first step. Now moving on to the second step. What is the second step over here? Let me just quickly mention. Oligonucleotide excision takes place. Now what do you mean by oligonucleotide excision takes place? That means the lesion that was initially present, it is now removed. In the previous slide, let me just draw over here. So let me just draw. So this is the original strand. Okay. Now in the first stage, I had shown you something like this. In the second stage, I said that the phosphodiester bond has been broken like this. And in the third stage, what is happening? The lesion has been removed. That means there is no lesion over here. So that is what is written over here. Oligonucleotide excision takes place. That means the lesion is removed. Lesion is removed. This is what is done. Now over here you can see after the removal of the lesion, the gap is created. So what is this? This is the gap created. Okay, now this is a gap that has been created and this gap has to be filled. Now which enzyme would be allowed over here, or which enzyme would be used over here to fill the gap? Obviously gap is created and it is repaired, and the gap is repaired by an enzyme called as DNA polymerase 1. I would be talking more about this enzyme in just a moment. And then finally, when the gap is repaired, something like this, let us say that the gap is repaired like this, then these bonds, the phosphodiester bonds, are then again sealed by ligase enzyme. So all of us know that the utilization of ligase is that it reseals the bond, or it remakes the phosphodiester bond. So this phosphodiester bond will be formed again. Okay, so I hope you got a clear understanding. I have a photograph over here, and this photograph is actually representing the base excision repair concept. Initially only I said that in case of base excision repair, the first step that we were using was the cleavage of the faulty base, or the removal of the faulty base. The faulty base was removed by a particular enzyme, and the name of the enzyme was DNA glycosylase. So this was the name of the enzyme. What it did was it was cleaving the faulty base. For example, you can see that this is a DNA strand. This is a DNA strand. And in this DNA strand, you can see that this particular molecule, or this base, is uracil. Of course, uracil cannot exist in DNA. It should be present in RNA only. So that means that this is a faulty base that is present over here. It should be removed instantaneously. Now, how would you remove this? You would use an enzyme called as DNA glycosylase enzyme, and DNA glycosylase enzyme would be cleaving the bond present over here. Remember that these bonds are glycosidic bonds. So the bond would be broken and uracil would be removed from here, and therefore this site would be called as apurinic or apyrimidinic site, depending upon whatever nitrogenous base you have removed. If you have removed a pyrimidine, then it is called as apyrimidinic site. If you remove a purine, it will be called as apurinic site. Of course, this site where the nitrogenous base is no longer present. So this was the concept of base excision repair. Why I was telling you? Because this is the point where the base excision repair and nucleotide excision repair are different. In the initial step of base excision repair, you are doing this. In case of nucleotide excision repair, you are not using DNA glycosylase. Instead of that, you are identifying the lesion and cutting the lesion on both of its side. That is what we are doing in nucleotide excision repair. So I hope you got a clear understanding. You can take a good photograph of this. Okay, so now let me move forward and discuss the next thing. So the best studied model for nucleotide excision repair, or the best studied example of nucleotide excision repair, is UVR system of excision repair. Remember, UVR system is just an example of this nucleotide excision repair. We would be studying this UVR system in detail in order to understand what are the enzymes that are used over here, so that we can get a better understanding about the nucleotide excision repair. Now, in case or with the help of UVR system or UVR excision repair, short stretch removal is done. Usually we can remove 12 nucleotides. That means if you have a lesion, if you have a problematic area of around 12 nucleotides, then you can majorly remove it by UVR excision repair system. So 12 nucleotides, this is an important point. Now, obviously, as the name suggests, we are again and again saying UVR. This should also be in small. UVR excision repair. UVR excision repair. That means there are three units over here, U, V and R. There are three units over here, three subunits over here. The first subunit is UVRA, second one is UVRB, third one is UVRC. So there are three subunits over here. And these subunits UVR A, B and C, obviously these are some proteins and they would be coded for some genes. So UVR A gene, UVR B gene, UVR C gene, they would be coding for some proteins and together they are called as ABC nuclease. Obviously these are proteins. ABC nuclease is protein. And since they are coded by UVR A, B and C, therefore together it is given a name ABC exonuclease. Now what do you mean by exonuclease? What do you understand by exonuclease? Exonuclease simply means that it would be used, it is an enzyme that would be used for cleaving nucleic acids. It would be used for cleaving nucleic acids. So this is the function of this ABC exonuclease. So now you can take a photograph of this and then we can move forward with the functionalities of UVR A, B and C in some very nice diagrams. Okay, I believe you would have taken the photograph. Let me move forward with the mechanism of UVR ABC exonuclease. So now UVR, what happens first, or how does this exonuclease system functions? So UVR A and B are attached to the DNA at the damaged site. Now, what is a damaged site? The damaged site is where the lesion is occurring. So, which two proteins they will be coming first? Always remember that UVR A protein and UVR B protein are attached to the DNA at the damaged site. So firstly, UVR A and UVR B will be attaching at the damaged site. I will be showing you an appropriate diagram also. First of all, let us understand what is the functionality of each of these components. The first thing is UVR A recognizes the damaged bases, or UVR A recognizes the damage. It is very important, it is most often asked in the examination that which system or which component or which subunit is responsible for identifying the lesion. So it is UVRA is recognizing the damaged. Extremely important point. Okay, now once obviously UVRA and UVRB came together, UVRA's function was to identify the lesion. What is the function of UVRB? I will be telling you in a bit. But now since UVRA has identified the lesion, suppose they have identified where is the problem, what will happen is that UVRA will be departed. That means the function of UVRA is finished now and UVRA will be sent off. Next, in place of UVRA, UVRC would come and bind. In other words, we can say that UVRC will replace UVRA. Replace UVRA. So UVRC would replace UVRA. Again, remember UVRB is still present over there only. Now, of course, after the departure of UVRA, UVRC has come along and now UVRC and UVRB are present and they would be occurring as dimer. So B and C together will be functioning as dimer. What will they do? We would understand in just a bit. You can take a photograph of this if you want. Okay, let us move forward. So now, what we are going to understand over here is, so UVR B and C have created a dimer. Now they will cleave, UVR BC, they will cleave the damaged DNA. Now basically the damaged DNA is cleaved by UVRC. This is the prime member or prime subunit that is involved in the cleavage. I will be showing you also. So they cleave damaged DNA, primarily UVRC, and what will it do or where it will cleave? On the 5' side, it will cleave at the 8th phosphodiester bond, and on the 3' side, on the 4th or 5th phosphodiester bond. What do I mean? I have shown with the help of a diagram. Now suppose let us say that this was the correct strand, the black one correct strand, and this the blue one is the faulty strand. Okay, let us say that this is the lesion over here, this is the problematic area over here. So on the 5' end of the problematic area, around 8th phosphodiester bond will be cleaved. 8th phosphodiester bond would be cleaved over here, and on the 3' side, approximately 4th or 5th phosphodiester bond would be cleaved over here. That means roughly this much area has been cleaved around the lesion. So lesion was over here, this area over here, and this area over here has been cleaved. So roughly 8th over here has been cleaved and 4th over here has been cleaved. So total how many nucleotides have been cleaved? 8 plus 4, and that would be roughly around 12. That is why I said that UVR-Xe nuclease or nucleotide excision repair majorly would be cleaving roughly around 12 nucleotides. Okay. Now, so this is clear. UVR-C would, again I am saying UVR-C would be doing this or cleaving this thing. It would be cleaving this phosphodiester bond over here. This is what would be done by UVR-C only. Now, UVR-D, what would UVR-D do? UVR-D functions as a helicase. Now, what does helicase do? Helicase, or in if you talk more specifically, then it will function like helicase too. What is the function of helicase? The function of helicase is to break the hydrogen bond between the two strands. Now obviously there will be hydrogen bonds that will be present between the strands. That means over here like this, the hydrogen bonds would be present, and helicase 2 or this UVRD, what would it do? It would cleave these hydrogen bonds. The hydrogen bonds would be cleaved off so that the lesion is completely separated. Lesion is completely separated. Something like this. If I draw it, then it would be more clear to you. So this is the original strand that I am talking about, and so this is one portion, and this is the other portion, and this is the other portion. The phosphodiester bond has been broken, and helicase also is removing the hydrogen bond are present over here, also, and the hydrogen bonds have been broken over here. Since the hydrogen bonds have been broken, easily this would be removed, or the lesion would be removed or cleaved easily. So it would unwind DNA, it will release the single strand between the two cuts. So this was one cut, this was one cut. Between the two cuts, the region would be cleaved off or removed. Now, obviously, then a gap would be created over here. Let me just rub it out and see if you are able to understand this. So, like this. So a gap would be created over here like this. So this is what? This is a gap. Again, if I want to repair this gap, if I want to fill this gap, I will require an enzyme, and the name of the enzyme would be DNA polymerase 1. DNA polymerase 1 is also called as Kornberg enzyme. Very important point. Kornberg enzyme. It is also called as Kornberg enzyme. So what will happen? The DNA polymerase 1 would be coming over here, and it would be filling the gap like this. Again, remember, DNA polymerase 1 is doing it, and then finally which enzyme will come? Ligase will come, and ligase will bridge these two bonds. Okay, so DNA polymerase has arrived, and then the next functionality would be of ligase. It will be bridging this or making this phosphodiester bond. You can take a photograph of this, and then we can move forward. Okay. So now, again, as I said, that DNA ligase will be utilized over here. So DNA ligase would be coming and gap sealing would be performed. Now, there are two very important diseases that are associated with the malfunctioning of nucleotide excision repair. That means if nucleotide excision repair is not present or it is not functioning properly, then majorly two kinds of diseases will occur. First is xeroderma pigmentosum. It has been asked in many examinations, so please remember this. And the other one is Cockayne syndrome. Okay, so these are the two diseases. Obviously, xeroderma pigmentosum, one question had been asked on xeroderma pigmentosum. In fact, many questions have been asked. So the children who nucleotide excision repair, if the nucleotide excision repair is faulty, generally infants or children are affected more, and the children that have this faulty nucleotide excision repair, they are not allowed to go and play in the daytime, because the sun rays can cause some mutations in the DNA, which can only be corrected by the nucleotide excision repair, since that is non-functional over here. So that kind of mutations might be fatal to these children. That is why they are not allowed to play during the daytime, and these children are allowed to play only in the night. That is why these children are also called as children of moon. Children of moon. Now, it has been observed that a lot of proteins are absent in people who are suffering from xeroderma pigmentosum. Actually, XPA to XPG, basically XPA, B, C, D, E, F, G, there are a lot of proteins, and XP basically means over here xeroderma pigmentosum only. So since the disease associated is xeroderma pigmentosum, the name itself has been given as XPA, XPB, C, D, E, F, G. So these proteins that are encoded at different stages of nucleotide excision repair, they are not present or they are not observed in people who are suffering from this. Okay. Now, so obviously they are present in the people who are having the functional nucleotide excision repair, and they are absent in the people who are not having nucleotide excision repair. Now, you can take a quick photograph of this and we can move forward. Okay. So this is a photograph that I have obtained from Levine's gene 12, and in this photograph you can clearly see what is happening. So this is the area of the deformation of DNA. In other words, you can call this portion as a lesion. So this lesion, first of all, is determined by, or UVRA and UVRB are the first two proteins that are coming to bind in this lesion, and the function of UVRA is to recognize the damage and identify the damage, and then UVRA leaves over here. You can see that UVRA leaves, and UVRA is replaced by UVRC. The UVRC is coming over here. You can see this is UVRC. UVRC is binding with UVRB, and the function of UVRC, as I have said already, that UVRC will be nicking on both the sides of the lesion. You can see that the nicks have also been made, nicking on both the sides of the lesion, and then finally UVRD would be coming. It would be breaking the hydrogen bonds between the two strands. In this way, the hydrogen bonds would be broken. You can take a photograph of this and we can move on to the next slide. Yeah, so as I said, so this is again another photograph of from the Levine cell book. In this, I am going to show you what kind of damages are repaired by nucleotide excision repair. The first damage, you can see that cyclobutane pyrimidine dimer is formed. You can see that this is one pyrimidine and this is another pyrimidine. So this is the phosphodiester. This is one chain of the DNA. This is another chain of the DNA. Obviously, the DNA is double stranded. So this is the first strand, this is another strand, 5 prime to 3 prime, and this is 3 prime to 5 prime. In this strand, you can see that the two nitrogenous bases, this is the first nitrogenous base, let me just say, and this is the second nitrogenous base. The bonding is being seen between the two nitrogenous bases on the same strand. This should not have been seen. Actually, the bonding should be seen between the nitrogenous bases of two anti-parallel strands rather than on the same strand. So you can see that 6 and 6, 5 and 5, they are basically bonding together, and this kind of cleavage is basically treated by nucleotide excision repair. So this is called a cyclobutane pyrimidine dimer. Cyclobutane, why are we calling it? Because meth, eth, prop and but, that means 4 carbons are involved. So you can see that 4, this is 1, this is 2, this is 3, this is 4. So 4 carbons are involved. That is why it is called as cyclobutane pyrimidine dimer. And then 6,4 photoproduct. You can see that the 6th carbon over here is bonding, is making a bond with the 4th carbon over here. So 6,4 photoproduct is also one kind of a damage that is corrected by nucleotide excision repair. In general, I have already said in the first slide itself that whenever you see or whenever you hear that a particular chemical mutagen is interacting with the DNA, then majorly nucleotide excision repair is used to treat it. At times, pyrimidine dimerization is also treated by nucleotide excision repair itself. So I believe you would have understood this. We can move on to the next slide. Now, this image is taken from Watson, the book, famous book Watson and Crick. Watson. Now, in this particular diagram, I would be showing you what is the function of UVRB. We already know about UVRA, C, and D, but how does UVRB function? It is written over here. I'll be telling you. Firstly, you can see that the distortion is present over here, or lesion is present over here. This UVRA and B, they come along together, and of course UVRAB together will be utilizing ATP also. They would not function without an ATP. Then you can see that UVRA and B, they are coming and binding to this lesion, and then identification thing is done by UVRA. UVRB will still stay over here. UVRA has left now, you can see, and then UVRC would be joining and it would be cleavage would be done. But what is the function of UVRB? The function of UVRB is it would be locally melting the strand. That means just around the lesion it will be causing the hydrogen bonds to break. This is a very important point. Local melting of the strand, or the hydrogen bonds will be broken at the lesion itself. Okay, but remember, UVRD was majorly responsible for breaking all the hydrogen bonds. It will be breaking the hydrogen bonds from far off also, but UVRB is locally melting. You can see that the hydrogen bonds are broken over here. So that is written over here itself. So you can see that the ATP hydrolysis promotes dimer formation by UVR, which forms complex with dimer B. UVRA and UVRB complex scans DNA to identify. UVRA leaves complex, and the remaining UVRB dimer melts DNA locally around the distortion. So this is the point that I was talking about. Finally, UVRC will be coming over here. It will be making cuts, and then UVRC would also leave, then DNA polymerase 1, and things would like that would come. Okay, so I believe you would have got a clear understanding with the different diagrams that I have shown. Now let us move on to the next slide, and these are basically the PYQs. I hope you would get a clear understanding of these PYQs also. It's very easy. The UVR ABC repair mechanism is involved in repairing, obviously, missing bases, strand breaks, cross-linked strand, DNA damage caused by bulky chemical adducts. Of course, this is the correct option. Option number 4, as I have already said this. Now, moving on to the next question. Genetic disorder xeroderma pigmentosum is due to the defect in. Xeroderma pigmentosum is a disease that is associated with the nucleotide excision repair mechanism. Option 2 is correct. Now, moving on to the next question. Okay, so excision repair system replace short stretch of DNA around the site of damage. The following statements are made about the nucleotide excision repair in E. coli. So we are talking about the NER, nucleotide excision repair over here. So which enzymes and things like that are being asked. UVRB homodimer creates the nick on one strand. UVRB homodimer creates the nick on one strand on both sides of the lesion. So this statement is not correct. UVRB does not create a cut. It does not create nick. It is UVRC that creates the nick. So this option is wrong. The 50 to 60 residue long stretch of DNA between the two nicks is removed by the action of UVRD. No, the 50 to 60 residue long, this is not the length of the residue. It is roughly around 12 nucleotides. So this is also wrong. The gap generated is filled by typically DNA polymerase 1. This is correct. The distortion caused by the lesion is recognized by and bound by UVRAB complex. True, this is also true. Recognition is basically done by UVRA, but the binding is done by UVRA and B both. So C and D options are correct. So third is correct for us. Okay, so these were the questions that I wanted to talk about in nucleotide excision repair. I hope you enjoyed the session. And just one more important thing, the GATB, if you are preparing, if you are an aspirant of GATB examination, the GATB crash course has started from 28th Feb, that is yesterday. If you are interested to join in the GATB crash course, you can connect with us on this number 8299134528. So this is the WhatsApp number. You can connect with us on this, and we would be telling you more details. And also you would can find out the video that we had made on this GATB crash course, and find out more details over there as well. You can connect with us over here as well. So if you liked the video, just hit the like button, subscribe to our channel, and also tell us which further lectures or which further topics you would want me to take. Thank you. Have a good day.